Hello, everyone, and welcome to the next installment of the Research Seminar Series 2021. And I'm Lucas Moutsianas, and I'm the head of Bioinformatics Research Services at Genomics England, where we work with researchers and enabling them, supporting them in using our data and the research environments to help answer their research queries. I would like to inform you, firstly, thank you for joining, and also inform you that the seminar is being recorded and it will be subsequently available on our YouTube channel. And as is the case with all previous seminars, this is a monthly series, and this is a seminar consisting of 2 talks, and then it's followed, every talk is followed by a Q&A session. I would please like to ask you to ask questions specifically using the designated feature, not the general chat, or alternatively, you can wait until the speaker has finished and then you can raise your hand and ask the question directly, verbally. I will talk to you a bit more about our speakers in a bit, but I'd like to mention that we want to showcase in this series the impact of the research happening in the research environments with the Genomics England data that, of course, very kindly and generously made available with the consent of our participants. And any researchers working on this data that would like to present their work here, please reach out to us, get in touch. So as I mentioned, this is research conducted using our datasets, which is accessible free of charge from individuals working in academic institutions. So academic researchers, please get in touch if you'd like to use this very rich dataset. And also commercial researchers can also get access to this data, and they can apply to join via our Discovery Forum. So please do get in touch, and you have the email address there on the bottom left. Yes, as I mentioned, please do get in touch if you would like to present in our future seminar and showcase your research to our audience. Before I move on to today's speakers, I'd like to remind you that this is a monthly seminar series. It happens on the last Tuesday of the month, and we're excited to have Professor Demian Smedley and Professor Anna Shu in the next one, which will be on the 30th of November. Talking about 100,000 genomes pilot on the rare disease diagnosis by Demian, and then by Anna on the non-coding landscape of chronic lymphocytic leukemia association with clinical outcomes. Feel free to follow us on Twitter to get further updates on talks. Finally, there is a number of initiatives that are taking place. If you would like to see all research that's currently happening in our research environments using this data, you can navigate to the research portal, and you can look all the projects and lay summaries. So do have a look if you'd like. Also, as I mentioned, follow us on Twitter. This is the handle there on any updates and research publications. Follow us on YouTube to have a look at previous research seminars as they're being recorded and uploaded. And finally, I'd like to bring to your attention the G Word, where our CEO, Chris Wigley, is talking with high-profile guests on all things genomics, as the title itself says. So do tune in and reach out to us for any questions. With this, I would like to move on to introducing you our first speaker, who is Athanasios Kousathanas, or Thanos, who is working with us at Genomics England as part of Bioinformatics Research Services, and has led the analysis on COVID-19, and that's what he will be talking to you about, our whole genome sequencing analysis on critical ill patients to reveal therapeutic targets for COVID-19. Thanos is a statistical geneticist. He has a background in population genetics and in Bayesian statistics. He obtained his PhD from University of Edinburgh, then did a research postdocs in Lausanne, in Switzerland, and the Pasteur Institute in Paris. And he's interested in developing a statistical methodology to analyze whole genome sequencing data in the field of complex traits. So over to you, Thanos, if you're ready.
What I will be presenting today, as Lucas said, is an effort that was led by Genomics, the Genomics Consortium, in terms of recruiting patients and also in terms of sequencing those patients by Genomics England. So this is work from a partnership between these two players. Next slide. So there's a lot of people that have contributed to this work, and I start with acknowledgements. Because of that, I'm highlighting the main authors such as Erola and also Kenneth Bailey from Genomics, and also from Genomics England, people that also contributed to this analysis. And most importantly, I want to thank all of the participants to the 100K Genomes Project, and also to all of the participants that contributed particularly for this study, for the COVID-19 project. Their contribution was absolutely essential for making these discoveries. Of course, also I want to thank all of the our funding from all of these societies that you see on the right-hand side, and also the COVID-19 Human Genetics Initiative, and also 23andMe that also contributed data for our replication. And with that, next slide, I want to talk about what is COVID-19 critical illness, because as I said in my title, what we are going to investigate in the study is basically the host genetics underlying the critical illness. And what we mean by that is inflammatory lung injury, which is mediated by the host immune system. And this can actually lead to very severe disease that sometimes leads also to death. And several factors, and as I mentioned, including host genetic variation, which is the objective of our study, influence the development of illness, which requires critical care or hospitalization following the infection with the virus. Next slide. So there has been a timeline since last year of discoveries in this domain. And it all started with the Ellinghausen last year paper in New England Journal of Medicine that discovered the first locus, the most replicated locus, which predisposes for severe COVID, and it's located on chromosome 3. Then Genomics also with I don't know if I can point. Genomics also with our aid as well published in Nature a paper that identified a further 4 genes on 3 chromosomes, and then also the Human Genetics Initiative, which combines data across the whole world, identified an additional gene, TAC4, on chromosome 17, which predisposes for critical illness, and another 7 genes that were for milder phenotypes. Their sample sizes were larger for milder phenotypes because there were some of the studies contributed to that consortium was basically based on questionnaires or just COVID positive tests. And we, as Genomics England, also Genomics, we are working with the Human Genetics Initiative together for combining our data sets and having new discoveries. Next slide. So what does it take to go beyond those studies and move forwards in the discovery of therapeutic targets? First of all, we will require larger sample sizes, which would be sample sizes of carefully selected cases. So our design works with selecting cases, as I will mention in the next slide, of patients that have been handpicked by clinicians rather than some algorithm that we impose on the data. And this is absolutely essential for the power of the study. The second bit is using whole genome sequencing and going up beyond and above what SNP genotyping can achieve by increasing the density of variants, interrogating rare and common variants, and also being able to assess multiple types of variants, such as indels and interallylics, that are beyond the scope of SNP genotyping arrays. Finally, we also use statistical methods such as fine mapping to distinguish and group independent signals, and also colocalization analysis to identify potential causal genes. Next slide. So our study's design uses whole genome sequencing at high coverage of all of the participants. Our cases are critically ill individuals with COVID-19 as assessed by the treating clinician, and the controls are from the 100,000 Genomes Project participants that Genomics England has sequenced previously, and also some participants, a small minority, that were recruited particularly for this study, and they have confirmed mild COVID-19, so they experienced only mild symptoms. Next slide. So you can see here the breakdown of our numbers for cases and controls. We have worked with four major populations: European, South Asian, African, and East Asian, as predicted, as we predicted their ancestry with genetic information. Next slide. And the first analysis, the major analysis that we did for discovery, was a genome-wide association study. And for that, we performed very careful variant quality control. We used only relatively common variants for this analysis with MAF above 0.5%. And we also performed, as you can see, several filters in order to eliminate potential false positives. We used a logistic mixed model regression as implemented in the method SAGE. And our covariates were sex, age squared, age times sex, and also 20 principal components to control for stratification. Next slide. Using this method, we can see here a Manhattan plot summarizing our discoveries. You can see on the y-axis the strength of, or the significant strength. It's minus log 10 p-value, which is basically standard for these types of analysis. And we have annotated the genes for which the variants that have the strongest p-value, the genes for which we have consequences, as our annotation software has indicated. And you can see here that we have 22 independent signals. And when I say independent, this was found with conditional analysis, which takes into account Likens equilibrium information. So all of the genes here that you see are independent signals. They are not, let's say, correlated in terms of the association. So these were found in the European population analysis. And then, next slide, we also perform a trans-ancestry meta-analysis. This means that we performed GWAS in each of the ancestries, and then we combined the summary statistical information with meta-analysis. And we found an additional 3 signals compared to the previous ones, and you can see here. And this is not surprising, of course, that it's only 3 signals, because our largest cohort was Europeans. So this means that we would have marginal new discoveries with other ancestors. However, it is important that we still found a few new loci. Next slide. Then when I talked about fine mapping, statistical fine mapping in the beginning, what we did after finding all these loci, and as you can see here, is that we zoomed into the loci, the significant signals that we found. As you can see here, it's a locus on chromosome 21. And we used an approach that has a sparse prior Bayesian approach that can identify, as you see with the black circles around the signal. So you can see with the purple dot, the top variant, and then with red, green, and other colors, other variants that are in linkage equilibrium with the top variant. However, not all of those are included in the credible set because of this sparsity in the variable selection that is induced, and then a certain selection of those variants is picked, and this variants are the ones that we say they belong to this particular credible set. And what this approach allows us, next slide, is to identify, as you saw, like, can you go to the previous slide? Yeah, as you can see, different credible sets that are like in very close genomic locations, but they are not in LD with each other. And we can statistically identify those different signals and then also follow them up with downstream analysis in order to narrow down even further the causal genes for the associations. This is something that is enabled by the whole genome sequencing that we have performed because of the density of the variants and because of the accurate assessment of linkage equilibrium without noise between markers. You can see, for example, here, two genes that are indicated on the bottom of the figure, IL-10RB and IFNAR2, that we believe are basically the causal genes in terms of this particular association, these particular two associations that you saw. And then a bit further away, there is, next slide, there is another signal there, which we have a couple of candidate genes for this one as well. Next slide. So the most important part of this study is what we call replication. And this is something that we can do because of other summaries that have been collected by consortia like the AGI that we also belong. And we can ask how many of the GWAS signals can we replicate in other data sets. And of course, replication is not as stringent in terms of the p-value requirement as discovery. And if you go next slide, and we found that 22 out of the 25 association signals were replicated using independent data from other consortia. Next slide. And when looking at other types of analysis that we can do, we also were interested to look also at rare variation, and we interrogated rare variants by collapsing those into a sort of burden testing type of approaches. And you can see here in this diagram that we can identify certain damaging variants, for example, missense or loss of function, and then we can collapse them into counts for cases and controls. Next slide. Then we can perform similar filters to the GWAS, however, with in this case with smaller sample sizes, because we were not able to filter noise sufficiently with the larger sample size that were coming from multiple types of sequencing platforms and also genotyping technologies. And when we performed this type of analysis, we particularly used two types of masks or types of mutations that we aggregated, which were loss of function and missense type of mutations. We used SAGE gene, logistic mixed model regression, and the test statistics were aggregated on per gene basis. Next slide. This analysis did not reveal any genes that were associated with COVID-19. Next slide. Then we also performed HLA haplotype analysis, where we imputed the major haplotypes of HLA, and you can see here that these particular haplotypes indicate different genes on the MHC region on chromosome 6. And for this particular analysis, next slide, when we tested each of the HLA loci, which you can see with different colors, we found a significant association with the HLA DRB1 gene. And this association was more significant than the actual individual variants, SNP variants that we assessed. And when conditioning on this association, the signal goes away. So we're pretty confident that this is the association that is driving the signal on the HLA locus that we saw previously. Next slide. Finally, we also performed a transcriptome-wide association study using the GTEx data. And this is expression data across multiple tissues. And when we performed this study, we also tried to colocalize the signals that were associated with COVID-19 with the GWAS signals. And this goes a long way together with what I mentioned before about the fine mapping and colocalization in terms of having a high resolution signal. Next slide. This analysis identified several genes that their expression, the expression change that is caused by the mutation that is associated with COVID-19 severity. In blood or lung, so the aberrant expression that is caused by this allele is associated with COVID-19. And you can see with the arrows the direction where the expression goes by the variant, the risk increasing variant, basically. So, for example, for NUC1 that you see there, you see that the expression is increased by the risk variant. So, for a particular clinician, a therapeutic treatment that would try to oppose that, for example, reducing the expression from NUC1, could be a therapeutic option. So opposing the changes that the risk variants are causing. So this particular type of analysis can help us a lot to basically find out how the risk variants are creating the pathophysiology of COVID-19, of critical illness for COVID-19. Next slide. So summarizing the results from all the analysis that I just showed you, we see an important role for interferon signaling, and the variants are predicted to result in a reduction in interferon signaling, and this is associated with the severe disease. Next slide. And I have two examples for which we have the actual crystal structure for the changes, especially for PLSR1, which is the exact protein. And the missense variant that causes COVID-19, that is associated with COVID-19 severity, is predicted to disrupt nuclear localization of a particular protein, and this is required for antiviral signaling and neutrophil maturation. So you can see there, like a particular bond that's basically disrupting the particular protein on the right-hand side of the plot. Then next slide. Then another example is a missense mutation again on IFNA10. And this mutation would replace an evolutionary conserved and bulky side chain on the hydrophobic core of the protein with a smaller one. And this would potentially destabilize the protein. So this actually is quite illustrative of the particular effect of these mutations on the immune system. Next slide. And we also identified other genes, as you saw, several other genes, and some words for those genes and their groupings is that they are involved in B and T cell development. Also lung disease, they have a lung disease involvement, and ABO histoblood group antigen secretion. And finally, as I mentioned, the HLA signal that is driven by a protective actually effect on HLA-DRB1. And just to mention that for this particular signal, there has been another paper in July, Langton et al. 2021, that also found a similar effects, but not in a genome-wide study as we actually replicate here. Next slide. So to conclude, the results are consistent with a multi-component model of COVID-19 pathophysiology. The failure to control viral replication and a tendency towards pulmonary inflammation and intravascular coagulation can predispose to life-threatening disease. And the discovered genetic associations of this study implicate new biological mechanisms underlying the development of life-threatening COVID-19, and several of which we believe are may be amenable to therapeutic targeting. Of course, large-scale randomized trials will be essential before translating our findings into clinical practice. Next slide. So there is a preprint that we published online in September the 2nd, and we welcome you to read more details on the preprint about the study and technical details as well. Next slide. And I just place a slide for the data availability. The summary data can be either obtained by Genomics, and the individual level data is available, and summary data will be available at the Genomics England dedicated cloud research environment. And I give you provide there the link to apply to become a GES member to be able to access the data with your project. Next slide. And of course, I will finish my presentation again, acknowledging all the people that contribute to this work, and also the patients and participants. Thank you.
Thanks a lot, Thanos. And we are now open for questions. Please use the Q&A button to submit them, or raise your hand so you can ask Thanos directly. I see a comment in the chat about the SL6A20 seeming to be a very interesting distinct gene. And then we also have a question from Said about how these exploratory results could help us in better diagnosis, prognosis, and treatment of COVID-19. If you can comment on that, Thanos.
Yes, as I mentioned in my talk, to go from the associations to actual treatments is a long way. However, we feel that our analysis suggests several genes several genes for which the variant, the mutation, changes the expression, and then particular therapeutic treatments that would try to ameliorate that could to target these particular genes could be tested in clinical trials. There has been some studies that try to work with replacing interferon signaling to provide interferons, for example, but these were negative. They had negative results. They were not actually better than placebo. However, this is something that, of course, is followed up by clinicians. And we are also in contact with many people that are looking at these results and are trying to find therapies.
Thanks, Thanos. Then we have a question by Menali Chan. Thank you for a great talk. Did you perform a specific gene set or pathway analysis on the GWAS data to try and identify specific functional pathways that might not have reached genome-wide significance?
What we did, we basically took several of the genes that we identified as significant, and we did the pathway enrichment analysis, I would say. And what we found was, I would say, several obvious pathways, such as interferon signaling. So this particular analysis did not reveal new insights, because already our results are quite targeted, especially in terms of the TWAS. However, we welcome follow-up studies to look more into that, as we have released all of the summaries. We will be releasing with a paper all of the summaries for people to look at them. Thanks
again, Thanos. A question by Obed Brew. I hope I'm pronouncing the name correctly. A great talk, Thanos. What transcriptomic data did you use, and are they available at the Genomics England research environment?
Sorry, I didn't mention many details there, but it was GTEx version 8, and yes, they are available. This particular analysis, actually, just to say, of the TWAS was performed exclusively in our cloud environment. So all of the resources were used within the Genomics England environment, in CloudOS. So someone could actually replicate it.
Thanks, Thanos. Given that João Almeida is asking, given that these variants seem to be causing problems to proteins that affect how the body reacts to viruses, how do we know that this is related, let's say, to COVID-19 only and not other viruses? And I guess we need more data from reactions to other viruses to figure that out. Comment on that?
This is an excellent question. Actually, we feel that our results could be actually useful for potentially other viruses or other diseases that are caused by infection with viruses, potentially. This, what I would say, that how someone could follow it up, if there are other GWASs for other diseases, they could check the genetic correlation between them and see if they can find the correlation of the effect sizes between the studies. And then this could actually be indicative whether they have a common underlying causality, like the same pathways are affected and so on. But it's a very interesting question, and we feel that it's very likely actually that there is common causes with other diseases, especially respiratory viruses.
Thanks, Thanos. Just to remind you to please use the Q&A specific function in Zoom if you can, although I will keep asking, of course, your questions from the comment section. We have a question from Philip Haycock. Thanos, given that this is a case control study, how do you know your findings aren't causes of COVID-19 incidents, essentially susceptibility, what causes people to catch COVID, as opposed to progression to severe COVID-19?
This is an excellent question. So actually, we are agnostic to what precisely is happening between. There are several stages, let's say, that our signals could have impact, including, for example, susceptibility and so on. But so our study is like enriched on people with critical illness. I mean, almost all of them have critical illness. So some of the signals could also be like signals for susceptibility. This is something that we have more data now with more mild individuals, and we'll be looking at other phenotypes. Like basically, let's say that it's just susceptibility and not critical illness in the future. But it is possible that, I agree with the comment. It is, we cannot necessarily say for sure, but I have to say that in our study, we're not like other studies that have like multiple cohorts together in their analysis. Our studies is really, the cases are like really critically ill. So this helps to make it more targeted in terms of the associations being highly likely causing likely, or at least associated with critical illness only, rather than something more mild. Because other studies have actually put together like multiple, let's say, patients, which some of them, do not have like as strong symptoms.
Thanks, Thanos. Samuel Smith asks, thanks for a great talk. Did your analysis identify any associations between ethnic background and COVID severity? We tested, this is an interesting question, we tested for heterogeneity across ancestries, and we didn't find substantial heterogeneity between ethnic backgrounds. However, there are differences in the allele frequency between different ethnicities. So it seems that some of the loci, of course, they might explain more variance in the disease, in different populations, because, of course, they're more frequent. So, in particular, there's a locus on chromosome 6 that is much higher frequency in South Asians than in Europeans. And the South Asians contributed most to that signal, actually. Thanks. Then we have a question from Konstantinos Varvagiannis, or rather split into two, I could say. Great talk. Did you have any signal for TLR7? Maybe I can ask it in two parts. First part of the question, did you have any signal for TLR7?
No. This is a very interesting question, because this in terms of the rare variant burden testing, but and other studies have found it, but I would say we replicate only marginally. We didn't find the gene-wide significant results for TLR7.
Great, thanks, Thanos. And the second question, part of the question, could you possibly comment on the transcriptome data, given a recent presentation at the ECG conference, that differentially expressed genes reflect disease-induced rather than disease-causing changes in the transcriptome?
I'm not aware of this presentation, Yeah, I'm not sure. Differential expressions different. I wouldn't say about causal. Yeah, I wouldn't be able to comment on the causality of this. So maybe we can touch with the person afterwards.
Sure. Let me have a look at more questions. Melanie asks a follow-up question. Did your analysis control for treatment, such as dexamethasone use, which have been shown to reduce mortality in COVID?
No, we didn't. We didn't take into account treatments yet.
Thanks, Thanos. Then Yoavo Ahonli, and I'm terribly sorry for the pronunciation. Thanks for the great talk. Do we have a special platform for all COVID-19 published works?
For all the published no, but the AGI, I would say it's the best resource for because all of the summary statistics go eventually there, including ours. So the Human Genetics Initiative online that also we together published a previous paper. They will have all of the data, all the summaries. For individual level data, unfortunately, each one has to go to each of the studies. This is like because of protection. Thanks.
There is a follow-up comment by Konstantinos. The presentation was from Dr. Porcu, University of Lausanne, from Dr. Raymond Group. So maybe we can have a look at that. Then we have a question that I think it's similar to one was asked before, but maybe it's worth reiterating from Mukasa David. There are other factors that predispose to the risk of developing the severe form of COVID-19 or critical illness, and this study includes only the critically ill patients as cases. How can we know if these variants are not also associated with the risk of infection, in which case the variants would also be found in the mild form of the disease?
Yeah, this is similar to the other question, I would say.
Maybe Thanos, would you like to make a follow-up comment here on your follow-up work with the mild cohort, where you looked into the mild cohort alone as controls?
Yeah, of course. Just to say that we also did an analysis just looking at severe versus mild only, and the results were consistent, entirely consistent in terms of the effect sizes. And this is something that has been found also by the AGI, like as well, when they compared the effect sizes between mild and severe. So I expect that there will be a high correlation between the two, a high genetic correlation. Perhaps the effect sizes are larger on the severe. There are a couple of loci, I would say, that could be indicating susceptibility rather than critical illness. So what this means is that yes, potentially the patients that have critical illness, they're also highly susceptible as well.
Thanks, Thanos. I think we're now done in the interest of time with questions. So thanks again for your presentation and for answering all the questions. And now we will be moving on with the second part of this seminar series, where we'll hear a talk from Professor John Bridgewater on prognosis and molecular characteristics of patients with track fusion cancer in the 100,000 Genomes Project. So welcome and thanks for being here. It's a pleasure. Professor Bridgewater. Professor Bridgewater is at the UCL Cancer Institute in London. He leads the National Cancer Research Institute in study group for upper GI malignancies, focusing on the clinical and translational research, has contributed to a number of landmark studies, and he's a chief and co-chief investigation on many of them in the NCRI upper GI research portfolio. And he also serves on several research committees, including the Clinical Trial Advisory and Awards Committee and the National Institute of Health and Care Excellence Committee. So welcome and thanks for being with us, Professor Bridgewater. Over to you.
Thanks very much indeed for that really very generous introduction, Lucas. I'm a clinician. That's basically what it says. I'm a clinician. So don't be too hard on me. So let me share my screen, which is what it says. It's the prognosis and molecular characteristics of patients with track fusion cancers in the 100,000 Genome Project. hopefully it will now. Here we go. Okay, so the NTRK gene encodes TRK fusion proteins, and these have been recognized to be driver abnormalities, driver alterations in a number of adult and pediatric tumors. It varies a lot, and part of this project was to find out to look at once again in a general population, look at the incidence of these genes, but of the fusions. But in fact, turns out that they probably end up being something like 1% for all solid tumors. There's a bit more detail later on. So larotrectinib and indeed its friend entrectinib from two different farmers, two actually very similar drugs, have been approved pretty much across the world, including by NICE, for use in adult and pediatric tumors with TRK malignancies, and the numbers are extraordinary. So if you have a TRK malignancy and you get given these tablets, and they're effectively side effect-free tablets, what we call the response rate, so the shrinkage rate, over 50% shrinkage in the tumor approaches 80%, and the survival is really extremely good. And this for an oncologist who's given chemotherapy most of his life is an extraordinary thing to have something that works unbelievably well and provides survival with really minimal toxicity. So in terms, this is where personalized medicine. So you hear this spoken of a great deal. Personalized cancer medicine. This is what this is all about, really. And and because it is so uncommon, 1%, and you will see less than 1%, there are very limited data on the natural history. And to use 100,000 genome projects to illuminate that a bit more was essentially the intention of this project called Voyager 2, funded primarily by Bayer, who make larotrectinib. And this was the algorithm about how, on the right here, about how we got there. So using the whole genome data, was there a NTRK 1, 2, or 3 structural variant? Was there a fusion? And then a number of things to in fact check that the calling of this fusion was correct. And you guys who work on the data the whole time will know about this much more, infinitely more than I do. These are all patients, something like 23,000 cancer genomes were sequenced, and these were all between March 2016 and 2019. And there were two steps to this. First of all, the control whole NTRK fusions were looked for in the whole tumor group, and then people who had more than one cancer were excluded. Other biomarkers. So we have a number of other actionable alterations here, which may have been interesting. Which are currently treatable in terms of cancers, were looked at, and the overall survival was in fact the primary analysis. The index date was the date of the initial diagnosis of cancer. So that's it so far. And so really this is the nitty-gritty, really. Here we have the demographics table on the left here. So the fusion positive group. So out of, in fact, 15,185 patients, there were 38 positives. Okay, 38 positives. And so if you compare these two groups in terms of demographics, they're actually extremely similar. The data in the right-hand table are the ones really of some importance. So these fusions occur in something like 2% of childhood cancers, brain and otherwise. And then in adult cancers, which is everything else apart from the top two rows, the incidence is below 1%. And in fact, in lung cancer, well below 1%, 0.06%. And you can see that it's actually what it says on the packet. This is a tumor. This gene fusions occur. TRAC gene fusions occur across all cancers, some more than others, but really it's a pan cancer phenomenon here. 0.06% of lung cancer patients. That is diminishingly rare, and you know, if we've got time, maybe we'll go into whether it's in some ways almost ethical to even consider the question. So here, this is the same information shown in a slightly different way. The most of these TRAC fusions were found in breast cancers and colorectal cancers, but that's purely because of the numbers of breast and colorectal cancers in the whole 100,000 genome analysis. Sarcomas, childhood cancers, and then, as you would expect, a tail of other things. Pretty much distributed. I mean, the numbers are very small, so you can't be absolutely sure, but a fairly even distribution between all three NTRK genes, perhaps a few more in NTRK3, but the numbers are so small. And then this is of some interest. 29 different TRAC fusion partners were identified. The most that any fusion partner, the highest frequency of any fusion partner was this ETV6, 4 out of 38. So this means this replicates the picture we see in other fusion abnormalities in other cancers. The TRAC fusion is extremely promiscuous and will fuse with virtually anything. I'm sure it's, of course, we haven't, and that's one of the things I'll suggest towards the end, is that, you know, does it fuse with everybody? You know, are the fusion partners anything? Are they co-located? Are they on nearby chromosomes and so on? So all of those data have yet to be examined. So I'm sure it isn't that promiscuous, but actually this tells us that the correct way of identifying these abnormalities is with RNA-seq or whole genome RNA-seq if you haven't got the luxury of fresh tissue. This is the other primary finding here. So they matched the fusion positive group to a similar fusion negative group and compared the incidence of these other suspects in the down on the left here, these other common driver alterations, what we call driver or clinically relevant driver alterations down on the right here. And this is pretty much the standard, so the first column here, pretty much the standard incidence of these abnormalities in the whole population. And you can see that actually it doesn't really happen in the gene fusion positive cohort. And that tells us, and this is both in the matched and also the sensitivity analysis, that with the exception of perhaps TNB high. And so that tells us two things. That tells us that these are genuinely single abnormality cancers. Okay, they're single single driver cancers, and that's really that to a large degree explains their huge sensitivity to TRPV1 drug. Doesn't normally happen in cancer, and the other thing, but the other thing it tells us, and I'm not sure we got to the bottom of this, is that they are rather more they coincident with high TMB and MSI. So a more MSI cancers. So 7 MSI cancers out of this population of 38 is way more than you'd expect for the whole population. So so there's something in that. I don't think that we got to the bottom of that entirely. I'm sure there's a very clever explanation somewhere. This is the overall survival analysis, and essentially the survival is similar, essentially similar to that of the NTRK fusion negative population. So you're looking at population risk survival here for patients with for patients in this cohort. So that this is incredibly good for a medical oncologist who's been treating cancers for over 20 years. This is astonishing. And a tribute to years and years and years of genomic research and the nirvana really of personalized therapy. What does this mean for patients? So just put relatively simply in a way that most oncologists, let alone patients, can understand. NTRK fusions are rare. The outcomes are excellent. Routine identification. So that's the key thing here, and we may touch a bit on that. A lot of biomarkers in everyday practice. In my clinic this afternoon, for instance, a lot of biomarkers currently are second thoughts. Actionable alterations are second thoughts. You have a patient, they come to you, oh, well, we haven't looked for this, let's look for it. That is an unbelievably inefficient way of determining whether somebody has an actionable alteration. And what we need to do is make it completely routine. And Genomics England, working with the hubs, have gone a long way towards doing this, establishing routine NTRK RNA-seq for NTRK in every single cancer patient. But it's got some way to go. But only if we do this will we find these patients and be able to offer them this treatment. And it is, as I say, a question of embedding this routine into NHS practice, and a lot of history to overcome here. But that's essentially what we must do. And the process of working with the genomic laboratory hubs, the GLHs, is really essential, as well as education of the local providers. So that's what it actually means for the patients out there. The conclusion: numerically higher risk of death in patients with NTRK fusions compared to those without, but it wasn't really significant. NTRK fusions rarely co-occur, so they really are single alteration cancers. There was high MSI and TMB. What that's about, we have to work out. And this suggests that they really are a group of single alteration driven cancers, and critically with a treatment out there. What's left to be done? Well, I mean, there was lots essentially. That was a pitifully small list of coincident oncogenic drivers, and all you will notice all genomic, no pathway analysis, no methylome analysis, nothing like that. So huge amounts more to do there. There's a lot of stuff out there around the importance of the breakpoint, where the breakpoint is in other fusions. We have no idea whether that's important in NTRK or not. There's evidence of we need more evidence of the transcription of these abnormalities using RNA-seq. That would be really useful. We haven't done any we haven't done anything to see what these things look like under the microscope. I mean, that's how we've been doing it since the beginning of time. And the relationship to the morphological appearances of these things, I mean, it's a nice project. And then, of course, second fusions. So once again, in other cancers with fusions, not infrequently, for reasons, again, which we entirely don't, well, I certainly don't understand, you occasionally get two fusions in a particular gene. And what that means, I think that's an extremely interesting subject. So Genomics England, thank you very much. Patients, families, investigators. And that was all I was going to say. Thank you very much indeed.
Thank you very much for your talk, John. And yes, we're open for questions. Again, please use the Q&A button on Zoom to submit questions. And if you don't mind, I can start with one, because you mentioned the fusion partners and the further research for breakpoints and the potential promiscuity of fusion partners. So I wonder if you can comment a bit more on that. For instance, are the numbers enough to give us a picture? And was the picture what you were expecting to see?
So the numbers are very small. I don't think we expected to see anything. And actually, correct me, I've given you the wrong impression. Have we done any work on the fusion partners? Not well. Just the list there. I don't know what we expected to see. I think the real importance of the promiscuity of fusion partners is in the diagnostic, is actually, curiously, a bit away from this, it's in the diagnostic process. So for other fusions, still the standard way of determining whether a patient's got a fusion is actually using a hybrid DNA test. Okay. And it's still out there, and it's only NHS, it's only GEL that's been driving for RNA-seq to do, and clearly, you're going to miss a huge number if you continue using DNA tests to try and find these guys. So the promiscuity question, actually, it's all about the testing. It's the science. I don't think anybody entirely understands yet. But it's this and other fusions. The correct, the best test we have at the moment is RNA-seq in all the hubs. Yeah.
Thanks. We have a question by anonymous attendee. Why did you choose these specific genes to look for additional driver variants in? Because those are the genes for which we have drugs, essentially. Those are the actionable genes that we have. So does it matter if you have another co-occurring gene? It may be nice to know, but we can't do anything about it because we haven't got any drugs. These are the ones for which we have relatively good drugs, not nearly as good as this stuff. This stuff is like way the best. Thanks. We have a question by Deb Lancaster. Thanks, very interesting talk. The low incidence rate could mean that blanket testing would sadly probably not be cost effective. Do you request tests on all your patients? And if not, how do you select them?
So I think that's brilliant. I think that's the whole point. We can't select them. If you go around selecting, arguably you should select children, but actually, even children, it's only 2%. No, the whole point is that you can't select them and you have to do it routinely. Otherwise, you will never, this reflex way of looking for a fusion is fundamentally, profoundly inefficient way of doing it. It has to be done. And cost economically, it's one test to fit them all. So you're not just looking for the NTRK gene, you're looking for fusion, you're looking for other fusions. Increasingly, fusions are really coming into play now. We've got drugs for BRAF fusions, we've got drugs for FGFR fusions. The list is building up. So RNA-seq to look for multiple fusions in all of these driver genes that you think are responsible for a number of different cancers across different organs. That's the way we should be going, really.
Thanks. We have another question by anonymous attendee. Again, going back to the fusion partner. Does the fusion partner relate to the cancer type, like the transcriptional specificity?
Really interesting. I'm not sure that it. I'm not sure that it does. I think it relates more to the gene. Okay, so for instance, I don't think it's been examined in any. Well, I don't think it's been examined at all for NTRK. But if you look at other fusions, these are the sort of nearby genes. FGFR, for instance, TAC and BIC mostly, and these are nearby genes. They're not notable for any function, as far as I know, for any functional importance.
So it could be the genomic organization. Thanks. I think these are for another question from the audience, so I will please submit more, and I will ask a bit, going back to the biomarkers that you assessed, and you saw that they would rarely, these variants would never co-occur with the fusions, the ones that you looked at least. And I was wondering, is this something we expect? Is this something that is across all cancer types, or do we not know yet?
Okay, so perhaps the sort of importance of that you can illustrate with a couple of examples, really. So there's this, where you have what would appear to be a single driver alteration that you can target. Look, in contrast, at something like standard colorectal cancer, where you will have about 3 or 4 coincident alterations, a mess of fusions, mutations, EIDs, a mess of all of these things, and all of them capable of driving the malignancy. So the chances of effectively treating colorectal cancer with one drug is diminishingly small. This is treatable with one drug. That's the importance of that bit.
Understand. Thanks. We have a question from Samina Rahman. Would the drug company, she thinks she said Bayer, consider co-funding testing, i.e. a model where universal screening for these fusion proteins would be incorporated into the drug costs?
So I'm afraid it's more prosaic than that. So what happened was that both Bayer and Roche, who produced the almost identical other compound, entrectinib, managed to get the treatments supported by NICE, the National Institute for Clinical Instruments. Once that happens, in the process of that happening is that they work out a cost, and included in that cost is supposed to be a testing cost. And so these things, so NICE work out these numbers. And so in theory, NICE would have costed the testing into the price of the drug. And but they keep the numbers from us, so we don't know how much it cost. They hide the numbers. The drug companies hide the numbers and the deals that they have made extremely carefully.
Thanks. Any other questions? Please submit them in the chat or raise your hand. We have a comment by Deb Lancaster, a follow-up comment. It would definitely have been the health economic model sent to NICE.
Yeah.
For the previous comment. Okay, so if there's no further questions, then let me check the actual chat as well to confirm that I haven't missed something. Yes. Then I would like to thank you. Thank you, Professor Bridgewater, again. Thanks, Athanasios Kousathanas as well. Very interesting talks. Great to see work utilizing the 100,000 Genomes Project data, and fascinating. Good news, I guess, as well, with respect to the fusions not co-occurring with the attested biomarkers. With this, I would like to thanks to everyone for joining, and to remind you of the seminar next month at the 30th of November. Please tune in for that as well. We'll have Professor Damian Smedley talking about the results from the 100,000 genomes pilot on rare disease diagnosis. And then we'll hear from Professor Anna Shu talking about the non-coding landscape of chronic lymphocytic leukemia. And close the seminar by pointing you back to resources like the research portal to look at all ongoing research in our environment, all the projects and lay summaries. Please have a look. Our Twitter handle again, where you can follow for any updates on publication and news. The seminar videos, where you'll find all previous one, and this one will also be added in our YouTube channel, and follow up interesting discussions from our CEO, Chris Wigley, and various guests on all things genomics. So have a look online and tune in for that as well. And yes, thanks again, everyone, for joining, and thanks to the speakers. Great talks.
Thanks very much indeed.
Thank you.